Mixing and mass transfer in bioreactors. 1. Having mentioned its importance, we'll now move on to further explore the importance of mixing and mass transfer in bioreactors. Mass transfer is the movement of mass from one location to another. In general terms, it applies to the conversion of one form of mass into another. For example, in a bioreactor, cells use nutrient medium to grow and produce an important biochemical, which can then be collected from the bioreactor. A high level of mass transfer will result from cells or microbes growing and dividing in optimal environmental conditions, and will result in a higher yield of product. It is important, then, that ideal conditions of temperature, dissolved oxygen, and pH are achieved before mass transfer effects are taken into account. One of the major factors affecting optimum mass transfer is a uniform distribution of nutrients within the vessel. This guarantees that each individual cell has access to nutrients, or that a particular enzyme has enough substrate to work on. If there were pockets of non-homogeneous material inside the bioreactor, if the amount of dissolved oxygen was not uniform, or if there were temperature hotspots or cold spots, then it is likely that some cells would not be able to function optimally. The agitation system in a bioreactor ensures that there is even mixing of the nutrients and cells in the vessel. But does simply increasing the rate of mixing or agitation proportionally improve the mass transfer? Let's look at the effects that mixing has on a bioreactor's performance.